At the end, please be sure to complete the assessment and evaluation survey. It will pop up on your screen once the Zoom meeting is finished. So let me start out by saying it's a pleasure to introduce Dr. Tekin, who is a professor of human genetics at at the Miller School of Medicine, and he's the interim chair of the John T. McDonald Department of Human Genetics. He is board certified in clinical biochemical laboratory genetics and provides clinical services for children and adults with suspected or diagnosed genetic disorders. Dr. Tekin is also the director of the University of Miami Biochemical and Molecular Genetics Diagnostic Laboratory, and he has a career-long interest in discovering and delineating genetic variations underlying rare genetic disorders with a special emphasis on hereditary deafness and undiagnosed diseases. We are very fortunate to have him here today to share his expertise. Thank you so much, Dr. Tekin.
Thank you, Lana, for the introduction. And also, I thank the Mailman Center for inviting me to share with you some of the recent developments in genetics and genomics. And I'm particularly happy that this talk is just 1 week before the 50th year celebration of the Mailman Center. So hopefully you're gonna enjoy listening to my talk. So as the title states, I'm going to be talking about genetic and genomic studies on rare and undiagnosed diseases or conditions. As you all know, when we talk about the etiology or causes of human diseases or human conditions that are impactful on health, we mainly divide them up into two. One is genetic factors, and the other one is environmental factors. In reality, there are few cases where genetics plays a major role with very little environment, and in some few conditions where environment is much more predominant. But in reality, for most of the patients, it's a combination of the two. And for the large portion of diseases, it's somewhat similar contribution. That's why the big chunk of the patients having diseases, they have both genetic and environmental factors involved. But for the purpose of this talk, I will talk mainly on the rare group here, where genetic causes play a major role with some environmental contribution. And when we look at the genetic causes in that group, which is this group, which is rare in general population.
Get me water too, or no?
So when we talk about this group, and also the differences between the genetic factors in this group and the larger population, we also see a difference within the genetic causes. Because in the rare group, we talk about very impactful mutations or changes in DNA that will have changes in other biological markers as well. And these impactful changes are either single, just one event, one mutation, or maybe two within the same gene. And this is often called monogenic or Mendelian, based on the inheritance patterns. And sometimes this change involves more than one gene, but still a single event, and it could be like one entire chromosome or pieces of the chromosome, either missing or extra. So it could be chromosomal or some genomic changes like deletions, duplications. And in some cases, it doesn't affect the entire body, but it affects certain cells in the body. But still, we are talking about a very small number, often one mutation that is super impactful on the function and leading to a disease. But when it comes to more common conditions, one gene, the effect of one gene is often not enough to cause a condition. And in most cases, what we talk about is like polygenic, a lot of genes, a lot of changes, variants in different genes, and also combination of environmental factors. So you can think of, hypertension, diabetes, many forms of cancer, and so on. So this is really a lot of changes in our DNA or other molecules. And there's a group in between where mutations in small number of genes, not many, and also environmental factors leading to some type of phenotype. So if we defined what we mean by rare disease, is that officially, this is from 1980s, the official description of it is that any condition that affects less than 200,000 people in the United States. So that corresponds to about 1 in 1,500 people. So what is important about rare disease is that even though a single condition or disease is not that common, when we put all this together, it affects a very high populations, like 8% of the population. And more importantly, and relevant to this audience, is that like 2/3 of these conditions emerge during childhood, and then they cause severe disability in at least a quarter of these children. And also they are important cause of deaths during childhood. A recent study showed that the annual economic burden is $1 trillion from these rare conditions. So this has a huge impact, not just the health of people and specifically children, also economically, it's a big burden. And finally, there are certain challenges for these conditions. The first one is very obvious because these are rare. So usually they are not included in the training or teaching at the medical system. So the specialists, even specialists, not just the primary care physicians, are not familiar with many of these. And these patients tend to travel from one specialist to another until a diagnosis is made, and often they remain undiagnosed. And since these are rare, and there's not much funding available to study these conditions, the underlying mechanism is usually unknown. And that's also related to the treatment or cure, which is not available for the vast majority of these conditions, because there is no financial interest of big pharma, big industry, and also the funding, like I said, is not that high. That's why these conditions are sometimes called orphan diseases. So how many of these rare conditions exist? There's no single answer, but we can get an estimate from the genetics world. And the reason is that since 1960s, a catalog was started to give a unique identifier to each distinct human phenotype or a disease. So it's called online Mendelian inheritance in men. And the current number there is around 8,000. And this is the minimum number of the rare conditions. Of course, there's more that is not included in this catalog because this is just the genetic causes. And the second is that the number is getting higher and higher every year. It was around 6,500 in 2010. So about 100 diseases are being discovered every year as rare disease and included in this catalog. And mainly the causes of all these conditions, the rare or even common too, is linked with biological variation. And when we say biological variation, it can include a lot of different molecules. So it's the DNA is one major one. but also the epigenetic changes, the transcription expression, which is transcriptome, at protein level, at metabolomics level, and so on. So these are affected by environmental factors and diseases occur. However, I mean, this is, as you can imagine, this is a field that will have like thousands and thousands of different data points, and these are not that easy to analyze. So that's why from the very beginning, the variation in DNA was always seen as at least a starting point and most impactful variation that can explain rare diseases. And the DNA variation, was first noticed many years ago when first the molecular methods were developed. However, I'm sure you appreciate that it's highly technology dependent, and that's why the characterization of DNA variation have been impacted by the advances in technology. And one gold standard for finding DNA variation is really reading the entire genome, or so-called genome sequencing. So we can read the A, G, C, and T in the 3 billion base pairs of our genome, the two sets of them, one inherited from our fathers and the other from mothers. And then we can identify these markers or variation. And the cost was a big problem from the beginning. So when the first draft of human genome was released in 2001, the cost of sequencing a single genome was $100 million. And as you see in this graph, right now the cost is less than $1,000, and then even $100 genome is technically being entertained at the moment. And this graph, I think, very nicely illustrates something happened towards the end of the 2000s and reduced the cost of genome sequencing very significantly. And that was the invention of next generation sequencing. So when we say DNA sequencing, we are talking about reading really A, G, C, and T. And the gold standard for many years was Sanger sequencing, which was invented by Dr. Sanger in 1970s. And this was like some labor-intensive technique that each position of a piece of DNA can be read incorporating some radioactive markers to it. Then it became, a little easier and somewhat automated with the fluorescent tags to the nucleotides. But however, one limitation was a big barrier, and that was the size of the segment. So Sanger sequencing can only read up to about 1,000 base pairs or 1,000 letters. So like getting 3 billion out of like 1,000 each time is super expensive, and also super labor intensive. That's why that was a big barrier, a big bottleneck. So what happened towards the end of 2000s is that a technology was developed, which was also called massively parallel sequencing. Instead of sequencing DNA or genome like 1,000 base pair at once, everything, the DNA was chopped up and then sequenced all at once. So this is called the next generation sequencing. But at the time, it was also very expensive because it was a very new technology. The instruments were being developed, and chemistry was really difficult. And since it was expensive to sequence the entire genome, a solution came, and that solution was temporary, but it was very effective. So instead of sequencing our entire genome, why can't we capture the most important parts of our genome and then sequence them? The reason is that more than 80% of human mutations hit these regions, and these are called exons. in each gene, we have exons, which are translated into amino acid and polypeptide and protein, and there are introns. So the introns are usually regulatory, but the exons are the real blueprint that will be turned into a protein. And the technique was named as exome sequencing, and actually it was whole exome, but it's definitely not the whole. So the exons are captured or enriched with a chemistry, and then they are sequenced. So with this method, instead of sequencing 3 billion, just sequencing maybe 40 million base pairs, we were able to get very important information. And the first big discovery was made in 2010 by discovering mutations causing a rare disease called Kabuki syndrome, using exclusively exome sequencing. So that really opened up an era in genetics, and I think we have seen a big revolution since then. So in 2010, we knew only 43% of genes, mutations in genes, explaining these Mendelian conditions, the 6,500. Right now, this number is more than 80%. So a lot of genes have been discovered since 2010 by using exome sequencing, and later whole genome sequencing, which now has become more affordable. Of course, you know, from the get-go, we played a role here at University of Miami. Just me alone in my lab contributed discovering these genes, more than 24 of them, and then also other investigators here contributed to this revolution. And here's an example from my own practice. So when this is about 10 years ago, when I was taking calls, I was asked to see a 12-year-old child who was supposedly diagnosed with megacystis microcolon-intestinal hypoperistalsis syndrome, or in short, MMIH syndrome. So this is a condition. It was first reported in 1970s, and as the name implies, these people get huge bladder, urinary bladder. They are usually born with it, and then they cannot tolerate feeding because they don't have enough peristalsis in the intestine, and the colons are super small because they are not used. So there were maybe around 100 patients reported at the time when we saw this child, and there was no clear inheritance pattern defined. Most cases were sporadic, and this was a little disease because they cannot tolerate feeding, and then they develop liver dysfunction, and they require multivisceral transplant to survive. So this was exactly what happened to this child that we were seeing. She got the multivisceral transplant, but she was doing quite well at that age. And then I think she had some other problem that needed hospitalization. So before this, we actually had some other successes for finding genes with exome sequencing. But in this particular case, we just got DNA samples from these 3 family members, both parents and the child, and we did exome sequencing, and we found a region where you're seeing the normal sequence here, and this is Sanger sequencing peaks that you are seeing. Each color and peak represents a different nucleotide, and in this case, we found at a position that the child was heterozygous. You see the 2 bands at the same position, meaning that she had both guanine and thymine at this position. And that corresponds to an amino acid change because it was a coding region, and the position of the 178 of this protein was supposed to be arginine, but it became leucine. So what is interesting about this gene was that this gene was ACTG2, which is intestinal smooth muscle gamma actin. So basically, just with running 3 exomes in this small family, we were able to find a highly promising DNA variant that wasn't inherited. That was de novo, a fresh mutation in the patient. and then was affecting a protein that is very important for the contraction of smooth muscle. So just by chance, a few weeks later, we were asked to see another patient at Jackson. And at this time, this was a male who was born with the classic findings of the syndrome. And we sequenced the gene, the same gene, and we found another mutation hitting exactly the same position of the protein. But at this time, this led to from arginine to cysteine. So I mean, I think you can imagine that this is very unlikely to be just a coincidence. Having two unrelated patients having two different mutations hitting exactly the same amino acid in the protein, and we did some functional studies and showed the impact. But the interesting is that the arginine 178 was so important for the structure of this actin protein, because it was making a bond with another amino acid, histidine, and then when it became leucine, that bond was broken, and cysteine, that bond was broken as well. So we were able to discover this gene to cause this MMIH syndrome as a dominant trait. And then about a year later, another study just confirmed what we said. And then now this is other genes were discovered as well, but this is still the most common cause of MMIH syndrome. So through the years, of course, the initial instruments were replaced with more advanced technology. So this is the instrument we still have in the lab, but we replaced that with the more automated, very much high throughput sequencer. This one, which is in use right now. So this is the reason that the cost is now much less. But the technology, of course, didn't stop there. So here is the newest technology that we just received, and in this case we can read very long segments of the DNA, and then we can discover many more variants that are not easily discovered with the little bit age technology, which is called short read sequencing. The new technology is called long read sequencing. So when I say whole genome sequencing, what we are doing really is that we chop up DNA into pieces, and then through the chemistry and on the instruments, we just read each segment. And once we read each segment, we map these sequences to a reference human genome. There is a composite human genome. It's called the reference human genome. And we identify all those changes, which are called variants. And then by using a filtering methodology and bioinformatics, we just filter out the variants that are not causal. And then in the remaining ones, we try to find, one or two, and sometimes a little bit more DNA variants that might explain the phenotype. So, of course, after the exome was used again and again, the genome became more affordable. And here's an example how genome helped us to find the cause or the variant. So this is a family that I had seen so many years ago. 3 affected children, 1 female, 2 males. They were all born with profound deafness. And these are the audiograms you are seeing. They also benefited from cochlear implants. So we did exome sequencing in this family, thinking that we should find the mutation, but we couldn't. And we finally did the whole genome sequencing, which is, like I said, much larger and look at everything. But that's not the only reason that the genome is superior to exome, because also in exome, you have to use some chemicals to capture the regions. And certain regions are very resistant to capturing. That's why genome is superior in those regions as well. And that's exactly what happened. We found a mutation in a coding region, which is an exon, that was missed by exome sequencing. The reason is that this was a very difficult region to capture. And this family had the mutation. So this was a deletion of a gene, a recently annotated gene, that all 3 affected members were homozygous and parents were heterozygous for it. So that actually is very consistent with autosomal recessive inheritance causing deafness in this family. We quickly looked at our other families with that. We found another example, and then we found 2 other examples with larger families having a different mutation. So like 3 different mutations in 4 families are highly suggestive that this is the cause, and in the small gene called MINAR2. But oftentimes, because the numbers are so small, it's not easy to confirm causality just by putting a small cohort. So that requires confirmation in animals and some functional studies. That's what we did. So we got the mouse model of this gene, and the knockout model does show here. So 2 weeks of life is when the mice start hearing, and you see the normal hearing thresholds here, the sensitivity, and this is the even when mice started hearing, they actually did have very severe hearing loss. But as they got older, that residual hearing also is gone. So it's like completely gone, profound deafness in older mice. So that's a confirmation from a mouse study that this gene is indeed the cause of deafness. And then when we look at what exactly is happening in the hair cells, which are receptors of the hearing, in the electron microscopy, you see that from the zoom out, they look not that much different. So this upper row is normal stereocilia, or the hairs of hair cells, and this is the mutant, and you don't appreciate too much difference. But when you zoom in, you see that the some row, the lower row of stereocilia is depleted. So that's probably the underlying, at least a contributor to hearing loss in these mice. But what is interesting with this gene is that so in the beginning, there is not much, even though they don't hear well, like you saw in the electron microscopy, they still have hair cells alive. And losing hair cells is a very common mechanism of hereditary deafness, because the hair cells die eventually from the stress coming from the missing a gene. And it's important because hair cells do not regenerate. When they are gone, there's profound deafness. In this gene, even though it's not common, it's not a common cause of deafness, what we see is that at early stages, hair cells are still alive. But as the mice get older, so this is normal whole mount cochlea, you are seeing 3 rows of other hair cells and 1 row of inner hair cells throughout the cochlea or inner ear or mouse inner ear. And here is the mutant at 14 weeks. So they lost most of their sensory cells. At this point, even if we give the gene back to these mice will not hear. But if we do it early on, then can hear. So there's a window of opportunity potentially for gene therapy or gene editing in this type of deafness, and that's something that we are still working on. So I think, by this time, I gave you examples of, the rare diseases and how genetic technology can help finding a diagnosis and potentially find some targets for treatment. So I will talk about a team effort to really capitalize on all these successes that happened in genomics to find a diagnosis in these very difficult cases. It's called Undiagnosed Diseases Network. It was established at NIH more than 10 years ago, and then expanded as a network about 10 years ago. And then 4 years ago, the second phase started, and we became part of this network. So we are the only clinical site in Florida and some of the neighboring states. There are 12 clinical sites. And in addition to clinical sites, there's a DNA sequencing core and also animal models where we generate some models from the animals to see if the causality can be confirmed. It also has a metabolomics core where some metabolic changes can be assessed. So this network is available to actually any patient or person. There's an online application process. It requires a recommendation letter from a healthcare provider. And then once we receive these requests, we review the medical records and then see if this particular patient or person will benefit from UDN evaluations. And once that stage is passed, we do very detailed clinical evaluations, mostly by bringing them patients to Miami and in some cases remotely. And then we get some samples to run studies, including genome sequencing, short and long forms, and in some other cases, some complementary studies with RNA, protein, metabolomics, and so on. And finally, we consider creating or generating some animal models for further understanding. So far, this program has had over 6,000 applicants from the country, and less than half of them were accepted into the program because they were felt that they would get most benefit from the program. And among the cases that have been evaluated, about a third of them had a diagnosis through UDN. This is very impressive number, I have to say, because the vast majority of these patients already had like whole exome sequencing or many extensive genetic studies done. So this number is on top of all the previous evaluations. And it's not limited to just genetics. There's some other like autoimmune conditions, sometimes environmental conditions are investigated as well. We just conducted a retrospective analysis within UDN cohort, and then we realized that that there's an overrepresentation of non-Hispanic whites in the cohort, and underrepresentation of some historically underrepresented minorities. And this is something very actively being addressed within UDN, and we are looking for enrolling more patients from the minority communities and increasing their chances to get benefit from this program. And more importantly, we looked at the successes between different ethnic and racial groups, and the minority groups are at least the same or even better chance of getting diagnosed through UDN investigations. That's another point that make us believe that we need to enroll more and more underrepresented populations into UDN. So I'm going to give an example of a UDN case to demonstrate how UDN works. So this was a 3-year-old African-American boy living in Jacksonville at the time of enrollment, and he had normal development until age 1, and then he started regressing and hypotonia. He wasn't ambulatory at age 3. The family history unremarkable, and he had got done so many investigations, genetic and non-genetic, without an answer to all this very severe presentation. So we had trio genome sequencing in this family, and then we were matching the variants that we saw with his presentation, and we noticed this variant in a coding region of a gene called PLA2G6. So this variant was inherited from the mother. And you're seeing, I told you that the next generation sequencing, we read pieces. So this is a piece that is read, and G just represents a change or a variant from reference human genome. So some reads he has G, some reads he has the wild type. So this position is heterozygous for this person. So this gene was known to cause some neurological conditions, and in particular, infantile neuroaxonal dystrophy 1 was very similar to what he has had in terms of developmental regression, central and peripheral nervous system dysfunction. So then we focused on this gene, and we found a variant in the middle of an intron. So introns are very hard to analyze. First of all, we don't have a lot of data to really filter out variants within the introns. I told you these are usual regulatory regions and they don't code for a protein or amino acid. So as you see here in this graph, this person, the child, is also heterozygous for a variant in the middle of an intron. So the T is the variant, and if there is no letter here, it means that it's wild type. So, and he inherited this from his unaffected father. So it actually, you know, it makes sense. He could very well be compound heterozygous, meaning one mutation in a coding region and one mutation in a non-coding region. And that's why, you know, the inheritance pattern of this disease is recessive. So that explains why he may have this presentation. However, it's not easy to solve because doing functional studies for a variant in the middle of an intron is not that easy. So by doing RNA sequencing from this child, first from blood and then fibroblasts, we showed very interesting outcome from this variant in the middle of an intron. So this is the exon, and this is the normal intron. And then the introns are spliced out. They are removed from the final messenger mRNA. So it's just exon, and that's how it is translated to protein. So introns need to be correctly removed, which is called splicing. And this position in this patient created a new splicing site in the middle of an intron. And it picked up another position within the intron, and it basically created a pseudo exon, which is not supposed to code. But this patient, this region is coding extra amino acids. But more importantly, there's a stop codon in the middle of this pseudo exon. So whenever the translation machinery hits a stop codon that is much earlier than its normal place, it triggers a mechanism called nonsense mediated decay and degrades the mRNA. So the end result is absence of the final product. And these type of mutations sometimes are called poison exon because it really poisons the entire function. And we checked the level of expression in this child. We saw that the expression, this is the child's expression, these are normal controls, is reduced. And we said this is most likely because of nonsense mediated decay that is triggered by this early nonsense variant. And we blocked the nonsense mediated decay with a chemical, and we saw that it's actually the expression is increasing. So it's very important. The reason is that if we can block this additional splice site within an intron, we can get a normal function of this copy of the transcript. And so in recent years, molecules called antisense oligonucleotides were developed for several conditions. And then you can design an so-called ASO specific to this mutation, and you can actually skip this pseudoexon and can get a normal gene and function. So that's very exciting. We are working with a foundation now to develop an ASO specific to this region, and then hopefully we will use it on this patient, and hopefully we will have benefit clinically to extend his life, because this disease is actually like it's a fetal disease that's going to cause death eventually. So that brings us to the another very exciting development that happened in the past 10 years in terms of using genetics to develop and utilize some medications, some drugs specifically targeted to these conditions. So as you know, the drug development takes so long time. And first you have to find a target, and then you have to find some molecules to really deal with those targets. And then you have to do all these clinical trials, first preclinical and then clinical trials, and be able to use them in patients. So genetics started showing its impact on this this journey by somewhat shortening after the target identification. So once genetic markers are found in conditions, rare or common, so then you can strategize really interfering with those markers and see you can get some clinical benefit from them. A recent review shows that there have been 40 such targets identified in the past 20 years, and that led to development of 47 FDA-approved drugs. And the vast majority of these conditions are in metabolic disorders, but the others are quickly increasing, affecting almost anything we can think of as a disease. And a majority of these therapies are biologic, like the enzyme replacements for inborn errors of metabolism. But the group with gene therapy and gene editing is like I said, is increasing. And oligonucleotides, like the example that I gave you, the antisense oligonucleotides are also increasing. So this is very important development, because like in almost every family and every patient, the hope really to get involved in these studies is that if there's anything that is going to be obtained at the end of these studies, and for many years the answer was no, but I think now we are seeing a light for many of them, and then potentially we will see an increase in these drug developments. So at the end of my talk, I'm going to give a little example of what like a real life example looks like. We don't have a treatment available yet, but so I'm going to show you how this might happen. So there's a rare condition called geleophysic dysplasia. This was first reported in 1971 in children with short stature, short the fingers and toes, and also some significant cardiac and lung dysfunction. So about 100 patients have been so far reported, and we know that like significant portion of these patients, these children, die before adulthood because of the complications of heart and lung and airway. The gene was discovered in 2008. It's called ADAMTSL2. Even though a gene is known, the function is like very largely unknown, the mechanism of the disease. The TGF-beta pathway was suggested, but it's not really confirmed by other studies. So in our practice, we encountered a child who had clinical findings of geleophysic dysplasia. And the name geleophysic means happy face. So because the facial features of these children looked like they were happy, that's why it was named as geleophysic dysplasia. So in this child, we found 2 mutations, heterozygous, meaning that both copies of this gene was mutated in this child. And both mutations are missense. So, so the arginine 61 becomes a histidine, and then alanine becomes threonine at position 165. So we predicted that these 2 mutations probably impact the folding of this protein. This protein is excreted from cell to the extracellular matrix, so it works in the extracellular matrix. And we thought that this is probably the cause, because the protein cannot be excreted because of the folding is not correct. And to confirm it, we first generated an in vitro model of each mutation, and this part in the bottom is the excreted amount of the protein, and this is normal amount in the extracellular matrix or extracellular space, and this is with 1 mutation, the R61, and this is the second mutation. So this actually shows us that both mutations impair excretion of this protein, but the 165 is more severely impactful on the excretion. Just to confirm it in the patient, we got the fibroblast, and you see that this is the extracellular matrix amount of a normal fibroblast cell line, and this is the patient. So it's missing, you know, the extracellular amount. So basically, at least at the very basic level, the disease is caused by less amount of protein in the extracellular matrix. So then I think the immediate, you know, reaction to this was that, can we find a drug that will actually push this protein more to the extracellular matrix? And this is done a lot now in rare disease research. Whenever there's a, you know, there's a potential mechanism and can be tested, can we repurpose the FDA-approved drugs and see if they're going to show any benefit? So that's what we did. So we got a library of the FDA-approved drugs, more than 2,000 drugs, and we developed an ELISA assay to check the amount of extracellular protein. And then that showed us about 100 of these drugs, you know, potentially can have a beneficial effect to increase the amount in the extracellular matrix. This is a very promising outcome. However, you know, I'm showing these as an example of, you know, I think the barriers, you know, along the lines, you know, how you have to overcome one after another. So there was no model to really test if any drug will show any benefit, because the mouse model, the knockout mice just die right after birth. So we cannot test any drug in mouse models. That's why, and this is the mouse knockout, and they mostly survive, you know, at most 1 month. So this is the knockout survival. So we had to generate the models, the knock-in models, which are the same mutations that our patient has, and see how they survive. And luckily, they actually do survive. And finally, after all these efforts of over a year, or actually 2 years, now we have a mouse model that actually survive and then can potentially be looked at if any drug will be beneficial. However, we still have to show the clinical phenotype to see if the mouse model will just show the similar findings to the patients. And that's what we did. So the mice, as we predicted, the 165 mutation is more severely impactful. As they get older, they cannot put enough weight, so they're smaller, and then they have significant heart and lung dysfunction, and then heart dysfunction. So finally, after, you know, I think 2 or 3 years of work, finally we have a model that we can test some drugs to see if we're going to see any benefit, and that's what we are doing. Actually, this morning, just before this meeting, I was at our lab progress meeting, and we are seeing the effects of these drugs in mice, but we have to follow like a long time. And finally, we can try maybe gene therapy can be beneficial in this model. So in summary, rare genetic disorders are not rare as a whole, and genomic tools are available to end the diagnostic odyssey and discover new genes and new diseases. Functional studies are absolutely necessary to understand the mechanism of these conditions and potentially develop some type of intervention. And this is becoming more and more available. And at the end of my talk, first I'd like to really thank my team, starting with the clinical group. So we have an expanding and very enthusiastic clinical team that has the clinical geneticists, our nurse practitioners, dietitians, and genetic counselors, and we are happy to see any patient or talk with you about any patient if you think genetic consultation or input is desired. We also have the diagnostic lab here. It's CLIA approved, CLIA certified, and we have a CLIA certified whole genome sequencing test available, and also many other genetic tests available. So if you need to get any test done in terms of genetics, please let us know. And also I'd like to thank the funders of these studies and and the Undiagnosed Diseases Network, and the NORD, which is the National Organization for Rare Disorders, and we are one of their centers of excellence here in Miami. That's all I have today, and happy to answer any questions. Thank you.
Thank you so much, Dr. Tekin, for such an interesting and informative presentation. If anyone has questions, feel free to put them in the chat or ask Dr. Tekin. And please remember to complete the survey that will pop up at the end of this meeting. Dr. Tekin, I can't really thank you enough. It was really a fantastic presentation.
Thank you, Alana, and thank you all for invitation again.